Can you hear me?
Yes, sir.
Okay. So, I think already 60 students is sharing this screen. Why? What happened to all others? It is already 8:15. The class is supposed to start at 8. So 10 minutes I will grant. So those students joining after 15 minutes will not get attendance. Okay? Today 2 hours I had to speak and there is no extra time for completing your topics. So while the number is 67. Okay. Some more students, please join. Okay, you can message your friends to join. Hello. So we will start with vestibular apparatus. Vestibular apparatus. Okay. Audition you have completed. Somebody answer. Audition classes are over.
Yes, ma'am.
Yes, ma'am.
So, the vestibular apparatus and cochlea. These are closely related. The bony structure that is enclosing the vestibular apparatus, it is known as vestibule. And the vestibular apparatus consists of semicircular canals, ventricle, saccule, and endolymphatic duct. The main function of the vestibular apparatus is posture and equilibrium. Okay. So, we will be discussing the topic under the following headings. First, introduction, then functional anatomy of vestibular apparatus. It includes the semicircular canals and its mode of action. The next is otolith organs and its mode of actions. Then vestibular pathway, functions of vestibular apparatus, then vestibular function test, then finally vestibular dysfunction. Coming to the introduction, you can see the picture. Robert Barrett, this period is 76 to 36, 1876 to 1936. He is a Nobel Prize winner in Physiology of Medicine in 1940. for his work in physiology and pathology of vestibular apparatus. So you know vestibular apparatus, it can detect the position and motion of the head and body. There are two sensory modalities present in the inner ear or labyrinth. So as we have told earlier, the labyrinth consists of cochlea for hearing and vestibular apparatus for maintenance of posture and equilibrium. These are subserved with the help of receptors. So, the labyrinth is located in the petrous part of temporal bone, and it consists of bony labyrinth and membranous labyrinth. So, in the cross section, you can see the bony labyrinth inside that perilymph, then the membranous labyrinth. It consists of the endolymph. So, on the right side, you can see the petrous part of temporal bone where the labyrinth is located. So, you can see the bony labyrinth filled with perilymph and the body labyrinth consists of the membranous labyrinth. It is filled with endolymph. And you can see the snail-shaped structure, that is cochlea. You have already studied the structure of cochlea, and it is concerned with hearing. And also you can see the vestibule. So vestibule is the bony part. And the vestibulocochlear nerves, that is the 8th cranial nerve. And you can see the vestibulocochlear nerve supplying the vestibular apparatus. So, functional anatomy, the inner ear or labyrinth consists of bony labyrinth and membranous labyrinth. Bony labyrinth called vestibule, and it consists of semicircular canals. So, the semicircular canals inside the bony labyrinth and the membranous labyrinth, which consists of utricle and saccule. This together constitute the vestibular apparatus. On the right side, you can see the diagram which explains the functional anatomy. That is, you can see the 3 semicircular canals. They are present inside the bony labyrinth. And you can also see the membranous labyrinth, which consists of utricle and saccule. So, utricle and saccule is present inside the membranous labyrinth. And the membranous labyrinth also consists of semicircular ducts and ducts of cochlea. And the cochlea, the snail-shaped structure, it is also included in the diagram. Okay. So, as I have already said, semicircular canals together with utricle and saccule forms the vestibular apparatus, which plays an important role in maintaining posture and equilibrium. So, the interconnections between the semicircular canals and utricle and saccule, and the saccule which is connected with the cochlea, with the cochlear duct. That is clearly shown in this diagram. So, the position of the three semicircular canals you can see in the diagram. The superior canal, you can see the superior canal, and in the lower part of the superior semicircular canal, there is an ampulla. And this ampulla is connected with the utricle. Then the superior canal, it continues with posterior canal, and the posterior canal is also connected through an ampulla to the utricle. And you can see the horizontal canal. The horizontal canal is the small structure shown here. It is connected to the utricle along with the superior canal. So you can see the opening of three ampullas, that is the superior, posterior, and horizontal semicircular canals is connected to utricle through ampulla. And you can see the utricle and saccule. They are together connected, and it has an endolymphatic duct. And this endolymphatic duct continues as endolymphatic sac. This endolymphatic duct, it is connected to saccule, as I said, and the saccule is connected to the cochlea through the cochlear duct. So that is the connections of the this semicircular canals, utricle and saccule. So, semicircular canals, they are tubular structures, 3 in number, and it is located at right angles to each other. So, there are 3 axes of rotation. One is vertical axis, second is anteroposterior axis, and next is transverse axis. And the semicircular canals, they are filled with endolymph. As I have said, the outside the semicircular canals, there is perilymph. Perilymph. Inside semicircular canal, it is filled with endolymph. So here you can see the anterior, otherwise called the superior. The superior is also called anterior, then posterior or inferior, then lateral or horizontal. These are the 3 semicircular canals. So, this is another diagram which clearly shows the semicircular canals, utricle, saccule, and cochlea. Now, the position of the semicircular canals when head is bent forward 30 degrees. So, the face is shown in the middle. So we are describing the position of the semicircular canals when the head is bent forward 30 degrees. So left side you can see horizontal with the surface, and right side vertical plane which project forward and 45 degree outward. So the posterior semicircular canals, you can see in the vertical plane which project forward and 45 degree outward. So the posterior semicircular canals, anterior semicircular canals, and lateral semicircular canals. The blue, red, and black, the circular structures. That is the position in the vertical plane which project forward and 45 degree outward. Then left side, the horizontal with the surface. The position is represented here. The blue, that is posterior semicircular canal, and red, that is anterior, and black, lateral. So left anterior semicircular canal and right posterior semicircular canals, they are called the synergistic pairs. Synergistic pairs. So pairs walking in the same plane are called synergistic pairs. So here you can see the left anterior, otherwise called superior semicircular canals. They act synergistically to posterior semicircular canals. So you can see left anterior and right posterior. They are synergistic. Then left posterior and right anterior, they are synergistic. Then next is left and right lateral semicircular canals. They are synergistic pairs. So for easy understanding, the red arrow is given. So left anterior, right posterior. Then right anterior, left posterior. And two lateral or horizontal. They are the synergistic pairs. So part where each canal opens into utricle is enlarged and is called ampulla. So as you have seen in the case of cochlea, the ampulla is also present in the utricle, and it contains the sense organ of semicircular canals called crista ampullaris. Crista ampullaris. It responds to angular acceleration or rotational movement of head. So the crista ampullaris, it responds to angular acceleration or rotational movement of the head. So the crista ambularis, it is represented as the red structure present inside the ampulla of the semicircular canals. The maculae and statoconia, they are present in the utricle and saccule. Okay. The utricle and saccule consists of maculae and statoconia. The crista ambularis in the semicircular canals in ampulla reach on inner wall. Okay. You can see in the ampulla, you can see the crista ambularis. Crista ambularis. You can see the ridge. And on the ridge, you can see hair cells and sustennacular cells. You can see the hair cells. Okay. The blue cells, hair cells. And adjacent to the hair cells, you can see the sustennacular cells. It is marked. In the lower part, you can see the sustennacular cells marked. Then it is surrounded by loose gelatinous tissue mass. It is known as cupula. You can see the cupula. Now, processes of hair cells are embedded in the cupula. So, the hair cells present on the crista of the ampulla. The ampulla has a ridge, and this ridge is having hair cells. The hairs of the hair cells, they are surrounded by loose gelatinous tissue mass. It is known as cupula. The base is in contact with vestibular division of eighth cranial nerve. So you can see the yellow nerve fibers. The base of the hair cells are in contact with the vestibular division of the eighth cranial nerve. So the hair cells on the crista ampullaris, they are the receptor in the semicircular canals. So here you can see the hair cell. The hair cell, you can see the elongated structure. The resting membrane potential is minus 60 millivolt. And there are 30 to 150 rod-shaped hairs. They are known as stereocilia. The short hairs, 30 to 150 rod-shaped short hairs, they are stereocilia. The large cilium, that is kinocilia. The large, that is longest cilia is kinocilia. Then tiplings, they tie the tips of each stereocilium with its neighbor and finally to kinocilia. So the tips of cilia, they are connected with tiplings, and finally the stereocilia is connected finally to the kinocilia. So the base of the hair, a hair cell, you can see it is supplied by the nerve fiber from the uh, vestibulocochlear nerve. That is the eighth cranial nerve. Okay. Have you understood the structure of the crista and the hair cells?
Ma'am. Yes, ma'am.
So now we go on to the uh, detailed structure and the potential changes happening in the hair cells when an action potential is produced. So you can see the type 1 cells and type 2 cells. And you can see the stereocilia. The short hairs, they are known as stereocilia. They are interconnected with tipplings. And finally, this tippling is connected to the gynoecilia. And these tipplings, they are in contact with the endolymph. And adjacent to the type 1 cells, you can see the sustentacular cells. They are the supporting cells. The hair cells, as you know, they are in contact with the afferent dendrite of the vestibulocochlear nerve fiber. And also you can see the efferent axon adjacent to the dendrite. And the base of the hair cell that is in contact with the perilenx. And when an action potential comes, there is influx of calcium, and the potassium will be going out of the cell. So there is movement of the tippling which is connecting the stereocilia with gynoecilia. So you can see on the right side, the gynoecilia is not connected with the stereocilia. And also you can see the afferent dendrite and efferent axon separately connected to the hair cell. That is in the case of type 2 cells. Now, about the genesis of action potential in afferent nerve, when stereocilia and gynoecilium bend in direction of gynoecilium, that is, tipplings get stretched. So, when the tipplings are stretched, there is opening of ion channels in taller neighbor. Potassium from the endolymph enter and there is depolarization. Membrane potential decreases to minus 50 milliwatt. Then this result in the bending of the cilia in opposite direction away from the gynoecilium and reduces tension on the tipplings, and this will close the ion channels and there is receptor hyperpolarization. So there is hyperpolarization of the receptor. So that is hyperpolarization occurs when the uh, stereocilia will be going away from the gynoecilia. This reduces tension in the tip links and causes the ion channels. So it will result in receptor hyperpolarization. So these ionic changes, they are shown in the diagram. Mechanical deformation away from the gynoecilia causes potassium channel to close. That is shown in the right side of the figure. So it will result in hyperpolarization and the impulses are going to the brain. On the left side, the mechanical deformation towards the gynoecilium opens potassium channels in the stereocilia and it causes depolarization, and you can see the calcium entering and the calcium will produce the the degranulation of the vesicles, and it will cross the synapse, and in the postsynaptic membrane, the depolarization is transmitted to the afferent axon. Okay. So left side, there is depolarization. On right side, there is hyperpolarization. From hair cells, appropriate signals are sent by vestibular nerve to CNS to appraise the changes in rotation of head and rate of change. So, the signals sent by the vestibular nerve, it will help the CNS to appraise the changes in rotation of head, how much the head is rotated, and the rate of change, that is the rate of rotation, in how much speed the head is rotating. Both these are sensed by the vestibular nerve and it is perceived by the central nervous system. So, the stimulus is angular acceleration, that is rotatory movement. In rotatory movement, one synergistic pairs of canals will be functioning maximally with respect to plane of rotation. Okay, so rotatory movement, one synergistic pairs of canals, that is left anterior with the right posterior. They are functioning maximally with respect to the plane of rotation. In any rotatory movement, stimulation occurs at the beginning and end of rotation. So, The stimulation is occurring at the beginning and end of rotation. The semicircular canals are oriented in three different planes and there are three axes of movement. Okay. Along transverse axis, nodding of head up and down. Then along anteroposterior axis, that is ear touching shoulder. So, in the case of head nodding up and down, that is the transverse axis. Then anteroposterior axis, that means ear touching the shoulder. In these cases, vertical canals are stimulated. Then along the vertical axis, that is shaking head from side to side, the horizontal canals are stimulated. So, along the vertical axis, the horizontal canals are stimulated. Both transverse and anteroposterior axis, that is up and down, and ear touching the shoulder. In both these cases, vertical canals are stimulated. Mechanism of stimulation and adaptation of receptors in semicircular canals. So, the first diagram, the direction of movement of endolymph away from the ampulla. That is beginning of movement. So, the endolymph is moving away from the ampulla. Then after 15 to 20 seconds, you can see in one semicircular canal, it is moving away from the ampulla, whereas in the other semicircular canal, it is moving towards the ampulla. Then when the head stops moving, the movement of the endolymph reverses. So, when the head stops moving, in the semicircular canal where the endolymph was moving away from the ampulla, there is the movement of the endolymph in the reverse direction. That is, it becomes towards the ampulla. In the opposite side, it moves away from the ampulla. Okay. The ampulla, you can see left side and right side. In diagram A, that is at rest. The diagram B, you can see direction of movement of endolymph away from the ampulla. That is beginning of movement. You can see in the left side, there is hyperpolarization. That is inhibition. That means the endolymph is moving away from the ampulla. Then after 15 to 20 seconds, you can see the reversal of the movement of endolymph on left side. You can see. Then when the head stop moving the same direction, that is towards the ampulla. The information received from semicircular canals during rotatory movement of head along three perpendicular axes is used by central nervous system to interpret the speed and direction of head movement and to make appropriate adjustments in posture and eye positions. So, according to the speed and direction of head movement, the central nervous system has to make appropriate adjustment in posture and head position. Posture and eye positions. So, during head movement, the information received from the semicircular canals according to the movement of the endolimb, the CNS uses this information to make appropriate adjustments in the posture of the person and also eye positions of the person according to the head movements. So that is about the semicircular canals. So the semicircular canals, the crista is the structure and the hair cells. Hair cells are the receptors, and hair cell consists of two types of hairs. One is stereocilia, other is kinocilia. And all stereocilia are interconnected, and these stereocilia is connected with the kinocilia. So, movement of the stereocilia towards kinocilia, it produces depolarization, and away from the detachment, it produces hyperpolarization. And that is about the production, the stimulation of the hair cells in the semicircular canals. Now, the semicircular canals, as you know, it is mainly concerned with detecting the angular acceleration. So this angular acceleration that is produced during the head movements. That is the movement of the head. That is up and down, then towards the ear and towards the ear touching the shoulder, and next in the vertical axis. So this rotational acceleration or angular acceleration is dictated by the hair cells and reaching the central nervous system through the vestibulocochlear nerve. These informations are used for the used by the central nervous system for making adjustments of the posture of the person and also eye positions of the person along with the movement of the head. So that is the main function of the semicircular canals. Then about the utricle and saccule. It is the other structure which constitute the vestibular apparatus along with the semicircular canals. There are two vestibular sacs. Utricle is the largest. It is connected to saccule by ductus utriculosacularis. The sense organ in the utricle and saccule, that is macula, and the receptors hair cell. And in the utricle and saccule, the sense organ is macula, and the sense organ is detecting linear acceleration or gravitational pull. So, the receptor in the macula is hair cell, and these hair cells are stimulated by linear acceleration or gravitational pull. Okay. So the sense organ is macula and the receptor is hair cell. In the semicircular canals, the sense organ is crista ampularis and receptor is hair cell. And this diagram shows the structure of the macula. The hair cells, sustentacular cells surrounded by gelatinous otolithic membrane. So here you can see the hair cells and sustentacular cells surrounded by gelatinous otolithic membrane. And otolithic membrane, embedded in it, there are crystals of calcium carbonate, and they are known as otoliths, otoconia, and statoconia. So, in the blue oval shaped structure, that is known as otolithic membrane. So, inside it, you can see the otoliths, otoconia, and statoconia. They are crystals of calcium carbonate embedded. So, the hair cells, you can see hair cells and sustentacular cells. They are present and they are connected by the afferent dendrites. The blue fibers, they are the afferent dendrites that is supplying the base of the hair cells. The process of hair cells embedded in otolithic membrane. Otolith more dense than endolith. The weight of otoconia bends cilia in the direction of gravitational pull. Arrangement of receptors in utricle and saccule. So as you know, the receptors are hair cells. So left side you can see the utricle. So utricle, the hair cells, and the hairs are embedded in the otolithic membrane, and macula is horizontal and hairs are vertical. So horizontally directed linear acceleration. So, utricle, it detects horizontally directed linear acceleration. So, right side you can see the saccule. Here the macula vertical and hairs are horizontal. Therefore, it detects vertically directed linear acceleration. That means gravitational pull. So, the horizontally directed linear acceleration, it is detected by utricle, whereas vertically directed linear acceleration, that is gravitational pull, that is detected by the saccule. The sensory structures present in the vestibular apparatus. There are 5 sensory structures, 2 otolith organs. They dictate the gravity and linear movements. So the 2 otolith organs, they are present in the utricle and saccule. Okay, otolith organs, they are present in the utricle and saccule. The utricle detects linear acceleration, that is in the horizontal axis, whereas saccule detects gravity, that is in the vertical axis. The 3 semicircular canals, they detect head rotation. As you know, the semicircular canals, the ambula consists of the consists of crista ambularis, and the hair cells present in the crista ambularis, it will dictate the head rotation, that is angular acceleration. So, vestibular apparatus has 5 sensory structures. The 3 crista ambularis and 2 otolith organs. Okay. Is it clear?
Yes, ma'am.
Are you clear how the
Yes, ma'am.
organs in the vestibular apparatus is stimulated? Okay.
Yes, ma'am.
The angular acceleration is dictated by the semicircular canals. The sense organ is, which is the sense organ in the semicircular canals?
Crystallis.
Crista ambulans. The sense organ in the semicircular canal is crista ambulans, and the receptors hair cells. Hair cells, that is stereocilia and kinocilia. The hair cells consist of the hairs, stereocilia and kinocilia. That is about the semicircular canals. Then about the utricle and saccule, they are dictating the linear acceleration, that is in the horizontal axis, and also gravitational pull, that is in the vertical axis. Okay. So the otolith organ. Otolith organ is the sense organ in the case of utricle and saccule. Okay.
Yes.
And also you have seen the depolarization and hyperpolarization occurring in the receptor cells. That is producing the impulse in the vestibular pathway. In the case of semicircular canals. So that is according to the movement of the kinocilia and stereocilia. Okay, related to the head movement. So the movement of the endolymph away from the ampulla and movement of the endolymph towards the ampulla. That is producing the movement of the deep links accordingly. Then in the case of otolith organs, that is present in the utricle and saccule. Here, a gravitational pull that is producing the movement of the hairs, that is in the vertical axis. It is produced in the saccule. Because of the weight of the autoconia, it produces movement in the vertical direction. So, gravitational pull is dictated by the saccules and the linear acceleration in the horizontal axis that is dictated by the utricle. Okay. This is because of the direction of the hair cells and position of the otolithic membrane. So that is about the stimulation of the hair cells in the vestibular apparatus. Now, how these impulses reach the central nervous system? It is through the vestibulocochlear nerve. The vestibular pathway. It is a three-order neuronal pathway. The nerve fibers arises from crista ampullaris and macula to form vestibular division of retina. So, first order neuron that is located in the vestibular ganglia. The other name for vestibular ganglion is Karp's ganglion. So, Karp's ganglion. So you can see the blue. It is represented as a blue bubble. Okay. You can see the vestibular ganglion. You can see the vestibular nuclei present in the vestibular ganglion. Superior, lateral, medial, spinal. These are the vestibular nuclei. The lateral is also called Dieter's nuclei. So you can see the vestibular nuclei, superior, lateral, medial, and spinal vestibular nuclei. So from the vestibular ganglion downwards, you can see the lateral vestibulospinal tract. So, from the vestibular ganglion to the spinal cord, that is the lateral vestibulospinal tract. And also you can see anterior vestibulospinal tracts, that is descending from the vestibular ganglion towards the spinal cord. And from utricle and semicircular canals, the impulses are reaching the vestibular ganglion, and from here it is going to the cerebellum. There are connections of the vestibular ganglia to the cerebellum. You know, there are vestibulocerebellar tract. Then you can see the superior vestibular nuclei. It sends fibers upwards. There are connections with the third, sixth, and fourth nerve. You know, the third nerve, that is oculomotor, fourth cranial nerve, that is trochlea, and sixth cranial nerve, that is abduction. These cranial nerves, they are mainly concerned with the eye movements. So, according to the movement of the head, we have to adjust the eye movements. Therefore, from the superior vestibular nuclei, fibers are going to the sixth, fourth, and third cranial nerves. So these are concerned with the eye movements in relation to the head movement. And these fibers are going in the medial longitudinal fasciculus. Medial longitudinal fasciculus. And from the third cranial nerve, that is from the medial longitudinal fasciculus, the fibers are also sent to the thalamus. Then, and the pathway is uncertain from thalamus to cortex. So, this is the vestibular pathway. The second order neuron, that is, it is located in the vestibular nuclei in the pondomedullary junction. The semicircular canals, otolith organs, then semicircular canals and utricle. So, the semicircular canals, it is controlled by medial and superior vestibular nuclei, whereas otolith organs, they are controlled by lateral vestibular nuclei, otherwise called Dieter's nuclei. the semicircular canal and utricle, that is controlled by the inferior vestibular nucleus. Uh, these are also called spinal or descending vestibular pathway. Then the third order neuron that is located in the thalamus, mainly postroventral and ventrolateral nucleus of the thalamus. So first order neuron present in the vestibular ganglion, second order neuron vestibular nuclei at the pontomedullary junction, and the third order neuron in the thalamus. So vestibular pathway, as you know, receptors are macula and crista ambularis. Then afferents vestibular nerve, center vestibular nuclei, and efferents. Efferents from vestibular nuclei. Efferents are vestibulo-thalamo-cortical tract. As we have seen, from the vestibular nuclei to thalamus and from thalamus to cortex. This pathway is uncertain. So the main efferent from the vestibular nuclei is vestibulo-thalamo-cortical tract. The other efferents, vestibulo-ocular tract. The next is vestibulo-cerebellar tract. The other is vestibulo-spinal tract. So, four efferents, vestibulo-thalamo-cortical, next is vestibulo-ocular, next is vestibulo-cerebellar, and next is vestibulo-spinal. The most important efferent is vestibulo-thalamic cortical tract. The function of the vestibulo-thalamo-cortical tract, conscious awareness of body position and changes. So the changes occurring in the body position, they are perceived by the central nervous system through the vestibulo-thalamic cortical tract. The second is vestibulo-ocular tract. Second is vestibulo-ocular tract. Here, the main function, movement of the eyeballs in relation to position of the head. So, the vestibulo-ocular tract so it controls the movement of the eyeballs in relation to position of the head. It passes through the medial longitudinal fasciculus, and it has connections with the third cranial nerve, fourth cranial nerve, and sixth cranial nerve, which are concerned with eyeball movements. The next is vestibulo-cerebellar tract. The friends passing from vestibular nuclei to cerebellum. The function is posture and equilibrium. According to the changes in the position of the head. It will make adjustments in the posture of the person. It also has the function of equilibrium because cerebellum, it has an important role in the equilibrium of the body, the vestibular cerebellar tract. The main function is posture and equilibrium. Finally, vestibulospinal tract. Vestibulospinal tract. It maintains tone in anti-gravity muscles. The anti-gravity muscles. Which are the anti-gravity muscles? The extensors. The extensors of the lower leg. The flexors of the upper leg. They are anti-gravity muscles. So, the tone of the anti-gravity muscles, that is extensors of the lower leg and flexors of the upper leg. The tone of these muscles are maintained by the vestibulospinal tract. Vestibular apparatus is concerned mainly with the reflex adjustments in posture and eye movements. These reflexes are called vestibular reflexes. There are two types of vestibular reflexes. One is postural reflex and the other is visual reflex. The postural reflex is concerned with balance and equilibrium during movement, and second, postural stability. The visual reflex, it is concerned with stability of visual images in spite of bodily movements. So, two types of reflexes, postural reflex and visual reflex. Postural reflex mainly concerned with balance and equilibrium during movement and postural stability. Visual reflex, stability of visual images in spite of bodily movement. So the postural reflexes, tonic labyrinthine reflex and labyrinthine writing reflex. The visual reflex, vestibulo-ocular reflex. So visual reflex, vestibulo-ocular reflex. Movement of eyeball in an attempt to fix at the center of retina when head rotates. It is sensed by semicircular canals. So the main function of vestibulo-ocular reflex is movement of eyeball in an attempt to fix at the center of retina. So fixation of the eyeball at the center of the retina when head rotates. So this is sensed by the semicircular canals. So vestibular ocular reflex, the response is head movement and it ends with movement of the eyes. Jerky movement of eye observed at the start and end of a period of rotation is called nystagmus. It is a reflex that maintains visual fixation on stationary points while the head rotates. So nystagmus. Have you completed the central nervous system examination?
Yes, ma'am.
Yes.
Yes, ma'am, we have.
Tested nystagmus in a person. No? Nystagmus. How will you test for nystagmus? So usually there are different types of nystagmus. The main type of nystagmus usually we test is lateral nystagmus, lateral or horizontal nystagmus. That is when we move the eyeball to one side, that is to right side. We keep the finger on right side of the head. Then ask the subject to move. Look towards the fingertips. So the both eyeballs will be moving to the right side. So you can, in the case of this cerebellar lesions, you have already studied about cerebellum. So in a case of abnormality of the cerebellum, when we ask the patient to look towards right side, there is movement of the both eyeballs towards right. So you can see jerky movements of the eyeball at the start and end period of rotation. So this is known as nystagmus. So this nystagmus, the commonest cause of central nervous system abnormality that is causes nystagmus is cerebellar lesion. Okay, horizontal nystagmus is present in the case of cerebellar lesions. This reflects so the cause of this nystagmus, that is in the abnormality of the vestibular ocular reflex. It is a reflex that maintains visual fixation on stationary points while head rotates. The latency of vestibular ocular reflex is 10 milliseconds. The pathway of the vestibular ocular reflex is shown in the diagram. The details are not necessary for you. So you can see the right and left vestibular apparatus. So the sacral, utricle and semicircular canals. So direction of movement is indicated by the black arrow. and the fibers from the right side, they are going to the left vestibular apparatus. The fibers from the right eye, they are going to the right vestibular apparatus. So, if you know about the fibers from the crossing of the fibers in the optic chiasma. Okay, the decussation, the at the optic chiasma, the optic tract, what happens? The medial fibers, it will be crossing. Medial fibers will be crossing, whereas the lateral fibers will be passing uncrossed. So this diagram shows the inhibition of the extraocular muscles on the other side and excitation of extraocular muscles on the same side according to the head rotation. So this is responsible for the jerky movements. The other reflex is otolith reflex. It initiates a reflex that prevents leg injuries when an individual walks downstairs or jumps from a platform. Otolith receptors responsible for this reflex are stimulated by linear acceleration of the head that occurs during descent. Now, functions of vestibular apparatus. First is maintenance of muscle tone. Second is maintenance of equilibrium. Otolith organs detects linear acceleration of head. Macula helps in dictating orientation of head with respect to gravity and helps maintenance of static equilibrium. Semicircular canals dictate angular acceleration. Now, maintenance of posture, this is by vestibular reflex. Then, orientation in space, it is mostly occurring due to the function of the vestibular receptors, visual cues, proprioceptive impulses, and cutaneous exeroceptors. All these receptors will send information through afferent pathway to the central nervous system for orientation in space. So the vestibular receptors also has an important role in perceiving orientation of the body in space. Then maintenance of posture. It is mainly through vestibular reflex. The semicircular canals, utricle and sacule. It has an important role in the tone, maintenance of tone and equilibrium. Now coming to vestibular function tests. The first is balancing test, one is straight line test, and the second is rhombus test. So, you know, straight line test. The person is asked to walk in a straight line, and after reaching a point, he is asked to turn back and walk in return. So, this straight line test usually in vestibular dysfunction, it becomes abnormal, and also in cerebellar dysfunction, it becomes abnormal. So the subject cannot walk in a straight line. Then rhombus test. What is rhombus test? The person is asked to stand still with the eyes closed. So, when the person is asked to stand still, when the eyes are open, even when the cerebellar function is abnormal, the equilibrium can be maintained through the visual input. So, and also the which is the case where rhombus test is positive. Sensory ataxia, that is posterior column lesions. The rhombus sign is positive in posterior column lesions. So which are the sensations that is passing through the posterior column? Posterior column sensations. ഏതൊക്കെയാണ്? Posterior column or dorsal column?
Fine
touch,
proprioception,
fine touch, kinesthetic sensation, two-point discrimination, stereognosis, that vibration, proprioception. All these are carried by the dorsal column or posterior column pathway. It's an ascending pathway. So, since it is the rhombus test is positive in sensory ataxia. That means the sensory information passing through the dorsal column, it is not normal in case of posterior column lesion. So when the eyes are closed, the person will be tending to fall. So this is known as rhombus test positive. Okay. So when the eyes are closed, the person will be tending to fall. This is a test for the posterior column lesion, and it is known as sensory ataxia. So that is the balancing test. So straight line test and rhombic test, it also detects the abnormalities of vestibular function. Okay. In cases of the vestibular uh, abnormalities of vestibular tract, the straight line test and rhomboid test becomes positive. Next is rotational test, that is Bereni chair test, and next is caloric stimulation test. Bereni, we have already seen the picture of the scientist, the Nobel Prize winner, who studied very much about the vestibular apparatus. So the rotational test is given his name, Bereni chair test. The next is caloric stimulation test. The next is electronystagmography. The Bereni test, subject is made to sit in the chair with head tilted forwards at 30 degrees. The chair is rotated at 30 revolutions per minute for 20 seconds. You can see the Bereni chair, the diagram on the right side. Effects: during rotation, the eyes open. The nystagmus occurs after rotation, that is post-rotatory nystagmus. Then dizziness, then vertigo, nausea and vomiting. So during rotation with eyes open, there will be nystagmus. In Bereni's test, the subject is made to sit in the chair with head tilted forwards at 30 degrees, chair rotated 30 revolutions per minute for 20 seconds. When the person is sitting with the eyes open during rotation, nystagmus occurs. Then the Berenice chair, it is stopped. After rotation, there will be post-rotatory nystagmus, dizziness. The person complains of dizziness, vertigo, and nausea and vomiting. Next is caloric stimulation test. Patient lies supine and head bent forward by 30 degrees. Vestibular stimulation by introducing hot and cold water into external auditory meatus for 30 seconds with a 5-second gap. The effects are post-rotatory nystagmus, dizziness, and vertigo. Now coming to vestibular dysfunction. 1. Labyrinthitis, Meniere's disease, benign paroxysmal positional vertigo. Benign paroxysmal positional vertigo. The next is motion sickness, and after labyrinthectomy also there will be vestibular dysfunction. Labyrinthitis means inflammation of labyrinth. Major symptom is vertigo associated with nausea, vomiting and dizziness. The Meniere's disease, abnormality of endolymph reduced by endolymph hydrops. Progressive deafness along with nausea, vomiting, dizziness. characterized by a triad of sensory neural deafness, tinnitus, and vertigo. Then BPPV, that is benign paroxysmal positional vertigo. Episodes of vertigo associated with staying of head in particular position occurs due to separation of otoconia from the otolith membrane. Next is motion sickness, excessive stimulation of vestibular apparatus, and the motion sickness comprises a collection of symptoms like increased salivation, abdominal discomfort, nausea, retching, vomiting, pallor, headache, and vertigo. So usually there's motion sickness. Usually it occurs when we travel by bus or train. Also in the air, uh, the travel by air also produces excessive stimulation of the vestibular apparatus. So, air motion sickness. This is also due to excessive stimulation of the vestibular apparatus. The labyrinthectomy means removal of labyrinth. The unilateral labyrinthectomy. Firing of impulse from one side. So it will produce nystagmus and ataxia. In the case of bilateral labyrinthectomy, positioning in space is lost if vision is disturbed. Then postural reflexes are affected, muscle tone decreased, and there will be hearing loss. So, to conclude, vestibular apparatus is responsible for maintaining balance, stability, and spatial orientation. Okay, that is about vestibular apparatus. Any doubt?
Ma'am,
Any doubt?
ma'am, ഈ സ്ട്രേറ്റ് ലൈൻ ടെസ്റ്റ് സെറിബുലർ അറ്റാക്സിയേക്കും ചെയ്തു നോക്കുമല്ലോ.
അതെ, സെർബലാർ അറ്റാക്സിയക്കും ചെയ്യും. ബിക്കോസ് സ്ട്രേറ്റ് ലൈൻ ടെസ്റ്റ് മെയിൻലി നമ്മൾ ചെയ്യണത് ബാലൻസ് ആൻഡ് ഈക്വിലിബ്രിയം ആണ്. സോ cerebellum, as you know, the cerebellum receives fibers from the vestibule through the vestibulocerebellar tract. So, in abnormality of the function of the vestibule, the coordination will be affected. Okay. So, both in cerebellar abnormality and abnormality of the vestibule or vestibular pathway, this straight line test, the person cannot properly perform the this straight line test. Actually, the cerebellar functions in central nervous system examination, usually we are examining the cerebellum. The vestibular functions are usually we are not testing. Okay.
Yes, ma'am.
or vestibular function. Usually, it is a specialty that is handled by the ENT. Okay, ear, nose, throat. It is a subspecialty. So, ear, nose, throat. So, we are performing the central nervous system examination. Here, the subspecialties, the special senses, that is ear, that the ear hearing, that is the tuning fork test. They are the main tests that we are doing to test the vestibulocochlear test. The other tests, they are the special tests, they are Berenice test and all others. They are not coming to our scope. So, we are mainly doing straight line tests to detect the cerebellar lesion. See, vestibular functions in our system examination, we are usually not testing the vestibular function. The audition we are testing, and in special cases, we will be testing this Berenice test and all. If we exclude this, we see a nystagmus, straight line test becoming abnormal. First, we suspect a cerebellar lesion. When the cerebellar lesion is excluded, then we test for the other tests, the vestibular function test, etc. Okay.
Okay, ma'am.
Yeah.
Yes, ma'am.
Any other doubt? What about space flight? You know, space physiology എടുത്താണോ നിങ്ങൾക്ക്? Space physiology എടുത്താണോ? Travel to space. Here also, there will be this vestibular dysfunction. When returning to Earth after a travel to space, there will be nausea, vomiting, etc. And this is mainly due to parasympathetic stimulation. And this is due to orthostatic intolerance phenomenon. Orthostatic intolerance phenomenon. Due to exposure to microgravity. You know, the person is exposed to microgravity. In the space, the gravity is decreased. So, it is known as microgravity. So, there will be baroreceptor dysfunction. Baroreceptor dysfunction. This will lead to orthostatic intolerance phenomenon. So, there is desensitization of baroreceptor, and this will lead to space motion sickness. This is also due to the uh, vestibular dysfunction. Vestibular dysfunction. So you know the gravity. When the person is exposed to microgravity, the linear acceleration, it is affected. It is dictated by the utricle. So it will produce a defect in the vestibular function. So space flight also will be causing the motion sickness. So this space motion sickness, it will solve after 48 to 72 hours after coming to the earth. Okay. So we will stop here. Anything?
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank you.
Thank you, ma'am.
Thank you, ma'am.